Chinese Academy of Sciences in Shanghai. Ling Ling is a really outstanding leader in the RNA structure field. So it is of great pleasure that we have her with us today. So Ling Ling currently has a lab in Shanghai, and she was also elected as a research scholar in the HHMI Medical Institute, International Research Scholar. And her work focuses on long non-coding RNA and the biogenesis and function of these regulatory RNA in different cellular contexts. And she has won numerous awards, more than I can ever describe here. So she is L'Oreal China Women for Science. She's on editorial boards of cell chemical biology, genome biology, etc. So it is really great pleasure to have her with us today. So Ling Ling, thank you.
Thank you. Thanks for the invite. And also my great pleasure to visit GIS virtually. And today I'll talk about linking RNA processing and function. Can you hear me well?
Yes.
Okay. So my lab started in Shanghai Institute of Biochemical Cell Biology about 11 years ago. And at that time, it was thought the vast majority of long non-coding RNAs is polyadenylated. So these RNAs just similar to mRNAs have their own promoters and are pervasively transcribed from intergenic regions and are polylated, except a couple of long non-coding RNAs, NIT1 and MALA1. So at that time, I happened to study NIT1 non-coding RNA when I was a graduate student with Gordon Carmichael, and found NIT1 is required for the paraspical formation, a type of nuclear bodies. More to me, importantly, it's a study from, or the insight from the early study in David Speicher's lab at Coastal Harvard. He found the superm of NIT1 and MALA1 are not polyadenylated, but are rather stabilized by triple helix structure. So this finding triggered me to ask the question, are there additional types of new RNA species that probably don't have polytails? So focused on this question, went back to my institute in collaboration with the computational biologist, Dr. Li Yang at the PSCD, a different institute from me, but in the same campus, we started to do the non-poly RNA sequencing. Aiming to identify the new type of RNAs. This approach allowed us to open up a new black box, I think, of the genome. So basically from this unconventional processed long non-coding RNAs, including the intron-derived snoRNA ended long non-coding RNA termed lncRNAs, or the circular RNA came from Larry introns and are negative with 2 and 3' phosphodiester bonds, as well as when others found those circular RNAs came from back-spliced exons. And later on, triggered by the finding of snoRNPs, we identified yet another type of long non-coding we call SPAR, which are derived from polycistronic transcripts and stabilized by snoRNA at the 5' end and 3' end are polyadenylated, came from those intron regions. So compared to this known long non-coding RNAs, all these new type of or unconventionally processed long non-coding RNAs do not have their own promoters, but are rather processed from Pol2 transcribed pre-RNAs and then stabilized by distinct mechanism, as I just talked about. So this earlier finding in my lab have led to the research directions in the last 5 years, including circular RNA biogenesis and function, as well as the link RNA processing and function in nuclear condensance and even during evolution, because some of those RNA we found are not evolutionally conserved. And also because these RNAs are completely, in the new formats, we have been trying to apply different but advanced technologies to understand their process and function, which in turn brings new biology and translational potential in biomedicine. And today in my talk, I'm going to focus on circular RNAs. So the single-strand RNA can be in the circular format was originally documented in 1976. Here shows the electron, the EM picture of denatured viral RNA from plant. This definitely is a circular or even like a tennis racket, okay? Those shaped circular RNAs. And then later on, it was found that group 1 intron self-splicing can have the circular intermediates. In the early 1990s, a handful of cases have shown that spliceosome can produce circular RNA with introns, but they were thought as aberrant splicing events with little functional potential. Now, with the application of the non-polyRNA sequencing, as well as the enzyme called RNAs, which digests the linear RNA and wide structure RNA, but enriches the circular RNA, followed by RNA-seq, we and others have found this back-spliced circular RNAs, as well as the circular RNAs from the intron layers. And today, my talk will largely focus on this widely expressed circular RNAs from back-splicing of exons or pre-mRNAs. This really efforts came from different labs have shown that they are broadly expressed in metazones, but generally at low levels due to the inefficient back-splicing efficiency. Most of the circles derived from middle exons of the genes. Therefore, the processing of circRNAs can inevitably interfere with the canonical RNA splicing, although at the low efficiency. And furthermore, it was shown that biogenesis requires spliceosome machinery and is modulated by cis sequences and transprotein factors because they came from exons. Once produced, they are largely localized in cytoplasm. But some of them can have the returning intron. Those can be retained in the nucleus and can regulate the POL2 transcription. Okay, and some others have been shown that they can compete with nucleotides to interfere with the signaling pathways and even sponge and scaffold micromolecules. So this is the overall picture of what we know about circular RNAs at the moment. And I'm going to talk about how we think about to address this novel class of circular biogenesis and function. So this is a typical gene that contains multiple exons. The canonical splicing leading to the formation of a linear RNA with all the exons are included. And back-splicing catalyzes distal splicing sites to forming the back-spliced circle. And in the meanwhile, circle-forming exons are excluded from linear RNAs. So the question was how the spliceosome can recognize its especially unfavorable reaction. We and others have demonstrated that the complementary sequences in the flanking intron of circle-forming exons, actually most of them are allo elements in primates or in human cells. They can form those transient pairing that can bring the distal splicing sites to close proximity, therefore facilitate back splicing. So the complementary sequencing in flanking introns in general are required for circular formation, but not sufficient for circular formation. That's because circular formation is regulated by the competition of unpairing within or across flanking introns. That said, if the unpairing occurs within this individual intron, then that can lead to just canonical splicing. But if the unpairing occurs across the flanking intron, okay, for example, this pairs with this, and then it can cause exon skipping and circularization. We have even a very simple plasmid system to demonstrate this hypothesis. Let's focus on plasmid 2 and 3. In the plasmid 2 containing one pair of complementary sequence can induce a high back splicing. However, introducing the competition over here, and then they can largely abolish circular formation. So because of this competition, we actually found another event that's called alternative circularization that said multiple circular RNAs can be produced from a single gene locus. Shown here is a famous gene called DMG3B, the DNA message transferase 3B. Computationally, more than 10 circular RNAs can be identified from this gene locus. And the highly abundant ones can be readily detected by denaturing page gel. Because of the widely distributed allo elements and the competition of unpairing human intron that lead to frequent alternative circularization. Indeed, more than 50% of the expressed gene loci, for example, in human ear cells, can produce more than one circular RNAs. So we were very basically amazed about this widely expressed circular RNA. So the question really is whether their formation have any sort of regulation and rules. Just here, we found at least 6 types of different alternative splicing events can occur in a final mature circular RNAs. For example, we found alternative back-splice site selection can be either alternative 5' back-splice site selection. So either can be these 2 exons or final circular RNA contains 3 exons. Similarly, also have alternative 3' back-splice site selection. And even within the final mature circular with multiple exons, we found this alternative canonical splice site selection, including cassette exons, means this exon can be either excluded or included in the final circular RNA product. Interrotation, either excluded or included, or similarly, alternative 5' and 3' splice site selection. So if you are interested in specific markers, wanted to ask how many circular RNA can be produced at region, we recommend this website in Dr. Li Yang's lab. Sorry for this. Okay, so it's collected more than 150 non-poly data sets that allow one to look at the circular RNA expression and conservation. So it's free for use. Okay, so obviously circular RNAs are widely expressed, except they are at a low level. But the question really is, what do they do? However, due to their circular conformation and sequence overlap with linear mRNA counterparts, make it very challenging to study circular RNAs at multiple levels, from annotation, validation, and loss of gain of function studies, because our difference between linear circular RNA at the back-splicing junction site. So we asked a fundamental question: if the cells already have the linear RNAs, why do they still make the circular RNA with almost with the same sequences except a different format? We wanted to study their biogenesis, stability, and conformation. Wanted to look at their entire life cycle from the linear RNA. Therefore, hopefully, we can get some new information about what circular RNA can do. So 2 students, Xiang and Chuxiao, worked on these very challenging questions. So previously, I have shown you the requirement of intronic cis complementary sequences for circular RNA. We want to ask how about trans factors. We had this question because we noticed that although having the same cis elements, expression levels of circular RNAs vary under different conditions. So we want to ask what trans factors are involved in circular biogenesis. So they made a very useful dual color reporter system that allow us to do the genome-wide screening for those factors. So basically, it's a reverse mCherry with the arrays that only the formation of the back-spliced circular can drive the translation of mCherry. And this entire cassette is put in the split EGFP and flanking by a pair of complementary sequence. So the canonic splicing and back-splicing forms the linear EGFP and the circular RNA that then led to the formation of the or translation of GFP and mCherry. And upon addition of Dox, we found the back-splicing events and also translation and also the translation of EGFP. And more than 99% of cells can produce at the same time both green and red colors. And we use the siRNA to knock down the circular RNA at the back-splicing junction site. We observe this noticeable reduction of the red color, but has no effect on the gray color. So suggesting these 2 channels are not interfere with each other. And then we use this system to go on human untargeted library screening and focus on this nascent circular level, meaning to identify the trans factor that probably regulated the circular biogenesis at an early stage. So using this screening, we have identified more than 100 factors that can potentially specifically affect circular formation. With those more than 30 cases reported factors in splicing regulation, that's as expected, because back splicing is one type of splicing. And we are expected to identify more than 10 hits related to viral and host immunity can also affect back splicing. And one of them is heterodimer of F90, F110 came from the gene called IF3. We found normally this heterodimer bound to the inverted allo elements that promote circular RNA formation. However, upon viral infection, these proteins are rapidly exported to cytoplasm, where they engage with the viral mRNA to inhibit the viral mRNA translation as one way to limit the virus propagation. In the meanwhile, we also noticed the global reduction of the nascent circular RNA upon viral infection. So this is the first evidence to show somehow circular biogenesis is not favorable during viral infection, and therefore linking circular biogenesis with immunity. And later on, they found not only at the nascent level, they found circular RNAs we know are very stable because they are not accessible by the linear RNA decay machinery, but they can be rapidly degraded upon poly-IC to mimic pathogenic upstream RNA for viral infection to be degraded with a half-life only within about 1 hour. And this is very specific to poly-IC because treatment with the cell of, to mimic upstream DNA stimulation or bacterial infection or even directed cell with inflammatory cytokines have no effect on circular stability. And we found the RNAs is enzyme responsible for the rapid and global circular degradation upon poly-IC or the virus treatment. So just want to remind you, this is a well-established picture of the RNA and innate immune system upon viral infection. So upon viral infection, the viral adaptation RNA can trigger the OS activation and then produce 258 with variable length from ATP, which can then trigger dimerization of RNA to make the inactive RNA become the active ones. And then that leads to the RNA degradation, including viral and cellular mRNA as the pathway, one of the pathways to inhibit the virus propagation. And then the adaptation RNA, long or short, or even the triphosphate sensing RNA can trigger those very famous immune sensing receptors, including TR3, MD5, and eventually lead to the activation of interferon and interferon-related genes. In the meanwhile, this viral adaptation RNA can also trigger the activation of another subgroup of nucleic acid receptors with direct antiviral activity, including PKR, F90, I just talked about, specific for PKR, which remains inactive in cells, but become active upon binding to the long double-stranded RNA of virus. And phosphorylate PKR can further trigger the phosphorylation of F2 alpha, led to inhibit protein synthesis. In the meanwhile, it can also trigger interferon activation. So in this well-established pathway, we found circular RNA can also be degraded by RNA. Importantly, we found the outcome of the circular linear degradation by RNA is different. So linear RNA, upon 6 hours of poly-IC treatment, we found about 10% loss. But for circular RNA, more than 90% of circular RNA are gone. So when we knock out the RNA, we see linear RNA now is no longer to be affected. Circular RNA affected by a little bit. This is actually because the reduction of the F90, because the export, the limit the nascent RNA formation. So nevertheless, the degradation outcome of linear circular RNA is very different upon poly-IC treatment. This is not because circular RNA is specifically or preferentially degraded by RNA. It's actually because back-splicing efficiency is very low in cells. It's a earlier work from my lab. We basically wanted to study how back-splicing is coupling with POL2 transcription. We found upon 4 hours of POL2, continued POL2 transcription, the splicing events become a plateau, but back-splicing can just keep accumulating. So back-splicing efficiency in cells is very low, but they are very stable. They can accumulate to high levels. So therefore, in the case of poly-IC and EMCV, the virus infection, so normally, let me say this way. So normally, the circular, although is slowly produced, but they can accumulate to high level. However, upon viral infection, they are rapidly degraded, led to the global reduction of circular RNA. So the question was, does this global reduction of circular RNAs play a role in immunity against the pathogenic adaptation RNAs. We actually analyzed the circular RNA structure compared to its linear RNA structure using the ship map with the chemical NI. To cut a long story short, we actually found those about 75% of the examined circular RNA tend to form this intramolecularly adaptation RNA region between 16 to 26 base pair long, and those structures are seen less frequently in their linear RNA cognate, and therefore they can allow the circular RNA as a group to band and regulate the PKR activation. So we found in some short adaptation RNA, some short circular RNA contain only one adaptation RNA region. Some long circular RNA can have even up to 4 adaptation RNA regions. Let me just briefly introduce you about PK activation. So PK activation requires adaptation RNA longer than 33 base pair. Shown here, incubated with the 79 base pair long adaptation RNA, the classical one has been used for PK activation, can lead to the striking PK activation. However, adaptation RNA within 16 to 33 base pair can block PK activation. Shown here, we added 33 base pair adaptation RNA at the same concentration as 79 base pair long that can significantly suppress PK activation. So now we replace this 33 base pair adaptation RNA with the in vitro synthesized circular RNA at the same concentration, a different circular RNA actually, they can suppress PK activation, but not the linear with the same sequences. So this suggests that circular RNA they can inhibit PK activation in a sequence but a structure in a sequence independent but structure dependent manner. In vivo, in cells, we see the same thing. So basically, we were able to detect the PK activation at 60 minutes of PKR of the polymerase stimulation. And this correlates with the half-life of circular RNA upon polymerase stimulation, 1 hour. And then now, when we introduce the circular RNA, all the linear with the same sequences to the cell, we found only the introducing of the circular RNA shown here, but not the linear RNA over here can block circular RNA, can block a PKR activation, okay? And this is not happening only in HeLa cell, but also observed in the mono cell like THP1 and the T cell ejected cells. So this suggests that endogenous circular RNAs are PKR inhibitors as a group. So stoichiometrically, we found more than 10,000 copies of circular RNA per HeLa cell, given they have more than one adaptation RNA region can form in some circular RNAs. So the real circular RNA can work as a PKR. The real element in the circular RNA can work as PKR inhibitor with even more than 10,000 copies. As another principal experiments, we knock out the RNAs. Now we found those RNAs depletion impairs this normal PKR activation shown here quantitatively, kinetics upon polymerase treatment. So this is one example of how we study long non-coding RNA. So basically, we studied how they are made and their life cycle. Specific for the circular RNAs, we found there are a group of molecules that are slowly produced. And once formed, they have those structures that allow them as a group to interact with PKR. And upon viral infection, they can be rapidly and globally degraded to release the PKR to be engaged with antiviral activities. And importantly, we found this RNA circular PKR axis is linked to the autoimmune disease, the lupus, SLE. Here are the patient samples from my collaborator from Shanghai Renji Hospital, Dr. Chen's group. So basically, we observed the elevated PKR phosphorylation accompanied with the global reduction of circular in all the 3 types of mononuclear cells, monocytes, B cell, T cells. I didn't show the data here, but they definitely globally reduced, as did from the SIE donors. And importantly now, when we reintroduce the circular, all the linear RNA to the patient-derived cells, like PBMC, the group of the cells, or only the T cells from SLE donors. Now we see the elevated PKR phosphorylation, as well as its downstream cascades including EF2 alpha, interferon beta activation, and interferon beta control gene expression. So this is actually suggesting an exciting potential of using cyclin as a potential therapeutic strategy for the lupus patients. Just wanted to say that those MIX1, Lin6E, and the IFIT3 are the well-established molecular gene signature of the SLE diagnosis. Although we're excited about the potential using of the circRNAs for targeting PKR with autoimmune disease, but there are still many questions remain to be addressed, including the exact molecular basis of circRNP interaction, whether we were able to see such misregulation of circRNP-PKR in the lupus animal models or other type of autoimmune disease animal models. And also definitely wanted to test the feasibility of modeling circRNA levels as a strategy for the lupus. After all, we wanted to ask this question: can we use the exogenous circRNA to dampen the RNA stimulated PKR activation? We had this question because there are also a few studies in the field have some controversial results in terms of the in vitro synthesized cyclin, whether they have the immunogenicity or not. So here shows one group actually from Harvard Chance group at Stanford. They basically made a very smart way to use to synthesize cyclin with T4 Fiji intron. And they found this exogenous cyclin RNA can potentially stimulate immune signaling via RIG-I. And the M6A modified endogenous cyclin basically can bypass cellular RNA sensors. In the meanwhile, another group from MIT, the Anderson's group, they actually found in vitro synthesized cyclin with anabola catalytic intron basically can diminish RNA immunogenicity because we know the linear RNA has the strong immunogenicity. So the question is whether unmodified exogenous cyclin can bypass the cyclin sensors. We think it's very important to address the question because in the future that can lay the foundation to use cyclin as a new type of RNA modality. Doesn't matter for translation or as PKR inhibitors. So Chuxiao and my student Siquan, they synthesized the cyclin based on this method, T4 or anabola or even direct T4 ligation. So basically, they were able to make this beautiful and clean cyclin. And they found with the T4 ligase, cyclin has no immunogenicity, while with the TD intron from Harvard's lab, accordingly, just has this immunogenicity as reported. However, the method 3 actually from anabola intron, we also were able to detect the certain type, level of the immunogenicity. And similar observations were found in different cell types. But nevertheless, we're able to make those immunogenicity-free cyclin RNA. Then we want to ask whether this type of cyclin without immunogenicity could suppress PKR activation in vitro and in cells. So basically, we compare to these known chemicals 2AP and C16. Those are two known small molecules that can inhibit PKR autophosphorylation. So came from the results that shows that those in vitro synthesized cyclin without immunogenicity can actually remarkably, suppress PKR activation with more than 100-fold activity. And similar effects were also observed in cells. And later on, we wanted to ask whether the cyclin could really bind to PKR or RIG-I. Shown here, this is in vitro shape map data with in vitro synthesized cyclin without immunogenicity. Again, we use chemical to do in vitro shape. And we found upon the PKR binding, we see the altered this bulge region close to the PKR binding upstream region, but not to other single-strand bulge region that distal from this PKR binding sites. However, compared to PKR binding, incubation of this circular RNA with in vitro purified regard didn't show much noticeable alteration of the shape map data. So further suggesting the PKR can bind to in vitro synthesize the circular RNA. So nevertheless, now we are working on the effect of the immunogenicity-free circular RNA on suppression downstream RNA-mediated PKR activation in animal models, and hopefully in the future we're able to use this circular RNA as a novel type of PKR inhibitors. All right, so although we have talked about circular RNA as a group can inhibit PKR activation that relates with autoimmune disease, we still wanted to look for individual circular RNAs with functional potential. However, methods at the moment are really, inadequate to discriminate circular RNA from mRNAs because they have the exact same sequence except the back-splicing junction sites. People in the field have used the CRISPR-Cas9 to do the exon depletion on certain circular RNAs, such as CRYAS. However, this is only unique case because this entire region is primarily produces the circular RNA in the neurons, but it's not useful for other cases because any knockout at the exon region then also affect the linear RNA expression. We had previously used the same strategy, but only to disrupt the intron RNA to block complementary sequence formation, therefore limited circular RNA. And we're able to reduce circular expression without affecting linear RNA expression. However, it's a very complicated procedure, and also depends on how many copies of parent can be formed within this circle-forming exon flanking introns. And many people have used RNA and shRNA to knock down circular RNA. However, half of the RNA is inevitably also targeting linear RNA that may cause off-target effects. So how to identify individual circular RNA with fundamental, with a functional potential? There's a key questions to address in the field. We're excited to see the report from Feng Zhang's lab about CRISPR-Cas13 system. And they also shown that application of the Cas13A in mammalian cells can effectively knock down linear gene expression. And as an RNA biologist, we were excited to see how to use the Cas13 system in the RNA biology study. So in addition of the knockdown, using this system, the catalytically inactive enzyme can actually also introduce RNA binding, tracking, and even editing. And also for this system, looks different from siRNA system because it shows the intolerant to the mismatch in the center seed region that hybridize with the target RNA. We actually wanted to ask whether such a system can allow us to adapt to use specific knockdown of circular RNA without affecting linear RNA using this system, targeting the back-splicing junction site. By the way, so for this catalytically inactive Cas13 system, we have done lots of screening, and we found the enzyme called the PSP Cas13B and PGU Cas13B can allow us to do the RNA labeling with only one pair of the gRNA get to the neat one RNA, for example, that allows to capture the paraspical furin and furin. So if any of you wanted to study abundant RNA labeling in living cell, probably can give a try of this PSP Cas13B system. Okay, nevertheless, I come back to circular RNA. So first of all, my postdoc Siqi, she actually picked up 9 highly abundant circular RNA using and then she designed the RNA that targeting the back-splicing junction site, 3 for each. She did, I think, maybe 2 or 3 shRNA for each circular RNA, and we found in general that can lead to about 40% of knockdown of the particular circular RNA, but with 15% of effect on linear RNA expression. and then want to use whether this intolerant to mismatch in the central cell region would allow to target the circular RNA without affecting linear RNA. So she basically designed the guided RNA targeting back-splicing junction site, even with 15+15 or 20+10 or 10+20, the spacer can came from 26 to 30 nucleotides long. And she did a screening of more than 8 different Ki-13 enzymes. And finally, he found the RFX Ki-13D can efficiently and specifically knock down the circular RNA up to 80% and without affecting its linear RNA expression. So here, 2 position matched SHRN and BSG gRNAs for each circRNA were assayed in 3 different human cell lines. Here shows more specific data. So he basically did lots of characterization, the mismatch, the length. Here shows the tiling of the gRNA spacer with the back-splicing junction site in the center exhibits the highest knockdown efficiency. Okay, this is red color for circular RNA and blue color for linear without affecting linear cognate RNAs. So why does the Ki-13 system work so well for circular RNA? Because on our hand, this system definitely works better on circular RNA compared to linear RNA. We think it's because at least 2 reasons. First of all, the overall conformation of circular RNA can be more rigid than that of linear RNA. So once targeted by gRNA Ki-13, such rigid structures in circular RNAs would continue to be targeted by this system, whereas in the linear RNA, they're highly dynamic and flexible, and so theoretically make it more difficult for effective targeting. And also because the back-splicing efficiency is very low, as I talked about earlier. So once cleaved by Ki-13, the newly produced circular RNAs cannot compensate for the loss of circular RNA, leading to the consistent reduction of the circles by this system. So nevertheless, we wanted to optimize this system to perform larger scale loss of function screens in human cells. To make the library, we want to first focus on those highly expressed circRNAs in multiple human lines that equals to more than 3 copies of circRNA, at least for 1 cell line. So overall, we put more than 700 circRNAs in the GRN library for in-cell screening. That represents the most highly expressed circRNA, more than 7% of them in examined human cell lines. And for each circRNA, we designed 5 BSG gRNA with different lengths. And for each of circle, we have its own paired control. So make it a very stringent screening. We did the circRNA screening for cell proliferation, 2 types of them. One is in-cell screening, another one is in vivo screening. So basically, we generated the RFX Ki-13D stable cell line, and then we put this lentiviral library, and then we pass the cell for 30 days and collect the RNA at day 0 or day 1 to day 30 for deep sequencing. For the in vivo screening, we have the RFX Ki-13D, the cancer cell line, and then we infect this library. And 7 days later, we put these cells into the nude mouse, and then wait for another 22 days for GRN library construction. So to make this work, okay, so basically after the screening, we have observed at least 3 different features of circular biology. So first of all, we have found cell type specific effects of circular on cell proliferation. So we found more than 60 circular in each cell line that show the effect on cell proliferation, but that drastically drop the numbers for circular in 2 or even 3 have the same effects in the cell proliferation in more than 2 cell lines. We were shocked about this result, so we have done lots of validations. Here shows you just some of them. Here shows the expression level of the linear RNA and also the parallel circular RNA in all the 3 different HT99, 293, and HeLa cells. So they are all expressed in these cell lines. And here shows the knockdown effects in all the 3 different cell lines. So they are all able to be knocked down to 70% in all the 3 different cell lines. And here shows a proliferation effect. So shows here, this is proposed circular RNA. Each of them has been used of 2 gRNAs, okay? So together we have 4 experiments. And we show this is specific for HT29 cell proliferation, but not for 293 or HeLa cells. Similarly, this is unspecific for 293, and these 2 circular are unspecific for HeLa cells. We don't know the mechanism, but this is what we observed. But we also identified the circular RNA, like circular RNA FAM128, as a common oncogenic circular that can promote that can promote cell growth. So here shows from in vitro and in vivo screening from HT29 cells, 8 circular RNA were overlapped. Among them have the circular FAM128. This RNA came from the linear RNA FAM128. It contains 4 exons. It's a cytoplasmic RNA. When we knock down the linear RNA, it shows the expression of the circular RNA and the linear RNA. So this linear is circular. This shows a knockdown of this particular circular RNA can actually shows this retarded cell proliferation in all the 3 different cell types. And similarly, we also observed the reduced tumor weight in the xenograft assay. So how does this circular RNA work? It turned out that this RNA promotes cell proliferation by upregulating its parental mRNA translation. Let me see. Yeah, so its parental mRNA FAM120 is an oncogenic gene related to the AKT pathway. So we knock down this FAM120A mRNA, that's also reduce cell proliferation. We knock down this circular RNA, didn't show the result, but it has no effect on linear RNA expression that show the retarded protein translation. So now when we transfect the FAM128, the linear RNA or expression vectors to the FAM128 deficient cells, shows here, let me explain to you, this is just non-treatment cells. This magenta color is treatment of the circular RNA FAM128 gRNAs that reduce cell proliferation, okay? Now with the overexpression of the linear RNA that can rescue the circular RNA caused cell proliferation reduction, okay? So suggesting it promotes cell proliferation by largely regulate its parental mRNA translation. So how will this happen? We look at the GIS very informative eClip dataset. We found circular RNA basically can interact with IGF-2BP2, which is a known translation inhibitor. In vitro assay, in the binding assay, we have shown that circular RNA, but not the linear RNA, can more specifically associated with IGF-2BP2, this purified protein. And in vivo RIP assay also shows IGF-BP2 can more specifically pull down circular RNA compared to other control circular RNA as well as the linear FAM128 linear RNA. Furthermore, in vitro assay also shows that circular RNA 128 outcompetes its linear isoform for IGF-2 binding. So shown here, this is with circular RNA. Now we should reduce the linear RNA bound with IGF-2BP2 without the circular, but as a control EGFP was added, that shows a stronger binding of IGF-2 with the linear FAM128 RNA. This is quantification. So we conclude that circular RNA 128 can prevent IGF-2, the translation inhibitor binding its covalent mRNA. Next question is the amount of circular RNA in cells sufficient for this proposed mode of action? We wanted to look at the stoichiometric ratio of these different players. So circular RNA FAM128 is one of the abundant circular, but still only account for about 22 copies per cell. However, the mRNA have more than 200 copies per cell. There are tenfold difference. the IGF-2BP2 is a multiple functional abundant proteins inside the cell. And we really have no idea in the very beginning how this circular could so specifically act on preventing its linear mRNA translation. Until we did the ribosome profiling, we found this circular 128 specifically enriched to the monoribosome. Okay, so here is IGF-2BP2. It's larger cytosol, but in all the different fractionations of ribosome fractions, GAPDH is only in the cytosol. For the different circular RNA, circular FAM120 is the one we studied on the monoribosome, circular RNA IgIPK3, which no micron sponge, is only in the cytosol. However, circular ZNF609 is known to be translatable. It's largely good and engaged with the polyribosome. Now, for the FAM128 mRNA, normally this green line shows it's largely engaged with the polyribosome. However, in the cells we treated with Cas13 gRNA 3 and 5, those two different gRNA to knock down circRNA FAM128. Now we see the significant switch of FAM128 mRNA locally distribution from polyribosome to monoribosome, suggesting this reduced translation as we observed, okay? And then meanwhile, the controls, the GAPDH and SP1, either normally enriched in polyribosome or normally enriched in monoribosome, did not show any effect upon circular RNA knockdown. So it's definitely knockdown reduced the FAM120 mRNA translation by affecting the FAM120 mRNA distribution on the polyribosome. So this is because this, so now if we count the subcellular localization of this circular and the linear RNA, we actually found the stoichiometric ratio between the linear, between the circular and linear RNA is actually now increased up to 1:3 and 1:5 ratio. Okay, so nevertheless, I have shown this data. This is another evidence to support how this circular RNA can reduce its mRNA translation. So this is a RIP assay with IGF-2BP2 pull down. Normally, the linear RNA does not really associate with IGF-2BP2, but upon the loss of circular RNA, now we see the increased binding between this FAM120 mRNA with IGF-2BP2. Okay, so next question would be, how does this physiologically, even still lower abundance of circular RNA acquire higher interaction capability with IGF-2BP2 than with the linear RNA? It turned out that circular RNA FAM120 is M6 modified, and then can recruit the IGF-2BP2, which is known to be M6 reader, to prevent its binding. So here, even here shows a different level of circular RNA modification that can increase its binding with IGF-2BP2 in vitro. And here shows that even at the physiological concentration, as low as upon 25 picomolar, we still observed the strong inhibition of the circular RNA. This increase the circular RNA modification, reduce the linear RNA FAM120 binding with IGF-2BP2. So this analysis suggests that an additional layer of translation and regulation of circular RNA, specifically circular RNA FAM120, that is enriched on monoribosome, where it prevents FAM120 mRNA from binding the translation inhibitor IGF-2BP2, eventually leading to the cell proliferation effect. So next feature of circular function is using this approach allows us to identify the circular RNA MAN1A2 as an important circular for most embryo pre-implementation development. So this set of experiments were done with my colleague, Dr. Chen Ling Ling, is a talented student Zhang Ling. So basically, she injected the alpha-capsid mRNA together with guide RNA to zygote and allowed the cell to develop into the blastocyst stage. And among 24 circular RNAs, this conserved circular we selected only one circular, MAN1A2, could significantly reduce the embryogenesis at the blastocyst stage. So here shows the individual knockdown of circmRNA-1A2 in the blastocyst stage, and here shows the cavity diameter that's done by after knockdown of this circmRNA-1A2. Importantly, now we implanted this circmRNA knockdown zygote into the pseudo-pregnant female mice that shows the retarded mouse post-implantation development in E7.5 days. So this is the first circmRNA that is important for the early development in mice. As I said, it's conserved circmRNA now even in the human ear cells. We knock down this circmRNA, we also observe retarded EB formation. So we're actually excited about this physiological function of the circmRNA. And hopefully in the future, we're able to dig out the molecular mechanism of how this circmRNA is responsible for this striking development phenotype. So the take-home message is the circmRNAs are well expressed, mostly in loci, but at low levels, and they can be regulated by both cis and trans factors. As a group, they can dampen activation related with immunity, and their misregulation is related to autoimmune disease. And I have shown you CRISPR after X Cas13D system can well discriminate the back-spliced circmRNAs from the linear RNAs, which will likely facilitate functional circmRNA studies. As you would tell probably already, it's very challenging to study circmRNAs because they have this circular conformation, they have sequence overlap with this linear mRNA except the back-splicing junction sites, but there are also excellent opportunities that allow one to explore new biogenesis and function and even potential application of circmRNA because they are different from all other type of RNAs. So now we know they are slowly produced, they have structures, they can be rapidly degraded. That stability would allow them to work as excellent biomarkers and even as circular RNA vectors, allow for circular translation, for example, and also their unique conformation may allow them to be to also have minimum immunogenicity that would facilitate to use the circmRNA as a new type of RNA modality. So fingers crossed for the circmRNA in the future. Okay, so this is basically what I have talked about today. Before I close up my talk, I want just to briefly summarize what we are working on in the lab. So earlier study in the lab, we use non-polyadenylated sequencing. We found this new RNA species. So recent studies focus on circular RNA biogenesis function and also lncRNA processing in nuclear reconnaissance. And of course, we have to use new technology to study them. And I want to mention that many circular RNAs and even snoRNPs, they are not evolutionarily conserved. Even the particular gene lockers is conserved, but the species-specific RNA processing produces a completely different RNA output in our human beings. So as an example, for example, last year we have published one study that shows that distinct RNA processing lncRNA can contribute to this non-conserved function stem cells. So I'm thinking probably in the future we have to use a different model organism to address the function of the circular and even non-coding RNA function. So with this, I'd like to thank the people in my lab who worked on circular RNAs, Xia and Chu Xiao, because of COVID thing. So they're still stuck in the lab, but they've been working on those fascinating stories. And Siqi, she's been my postdoc for 3 years. Now she's moving on as an associate principal investigator in my institute. So this is a picture from my lab in 2 years ago already to Disneyland. We are hearing, but before I give this to you, I wanted to thank my colleague Li Yang for also computational biology, Nanshen for those precious SLE patient samples, Yangming and Jin Song for mouse biology. Also thanks to the funding agencies. So thank you all for listening. I'm open for questions.
Thank you, Ling Ling, for the wonderful talk. So we have quite a few questions, actually. So, okay. So first, hi, I'm wondering if the current circular RNA datasets, more than 150 of them in one of the slides, is sufficient for the use of machine learning to predict the biogenesis of circular RNA computationally?
You excuse me, would you please repeat the question again? I was lost a little bit.
Oh, no problem. So I think they're asking if the current circular RNA dataset, I think he wrote there about 150 of them. Are they sufficient for the use of machine learning to predict the biogenesis of circular RNA computationally?
That's actually a good question. I don't think Lee is working on that. This is because circular biogenesis is quite complicated. In addition to the cis complementary sequences, they also have to be engaged with different transfactors. If we look at this data set, it's not only from human beings, it also came from, for example, flies, worms, and there's different organisms. And different organisms have different sound elements. I have the data, I didn't have time to talk here. We have found that through, through the evolution, there are more circular RNAs can be detected in human compared to the rest of the organism, even with the rodents. So that being said, it's probably quite difficult to simply use the current annotated circular RNA sequences to do machine learning, given this very complicated biogenesis pathway. Yeah.
Okay, thank you. And then are circular RNA splicing occurring mostly in the nucleus? What are the cues for nuclear translocation?
Good question. So there's only one study talking about the circular RNA transport from Jeremy Willis' lab. Beautiful study. So basically, they found the circular RNA, the short and long ones, use different hyperprotein to be translocated from nucleus to cytoplasm. But I believe that more proteins are, of different pathways are coming because in that study, they largely focus on using the fly SS2 cells. So again, this is a very immature field. So more studies are welcome. Yeah.
And then how do cells deal with wrongly spliced circular RNAs? Do you find any mismatched bases in circular RNAs in your datasets?
It's actually very difficult to do because any sequence you find in the dataset, except the long reads, you expect to see it can come from linear RNA or from circular RNA. If that reads happen to have the back-splicing junction site, and then you can discriminate from the annotation. Otherwise, you won't be able to see or to confirmatively say it came from linear or came from circular. So this is how challenging to study circular RNAs.
Then if you introduce linear RNA controls that are engineered to fold with double-strand regions similar to what you saw in most circular RNAs, does that also lead to inhibition of PKR activation?
Good question. So PKR activation, so I actually largely simplified the PKR pathways. So in addition to the circular we talk about here, there are even the snoRNA intermediates, other type of small RNA. They can also report as PKR inhibitors. But specific for your question, if you put this in linear RNA, but you have to deal with the ends of the linear RNA, okay, because it depends whether triphosphate or hydroxy group, so that can cause different immunogenicity. Yeah, so basically, I think the Dox-inducible region is essential to block PKR activation, not the circular structure. But forming the circular RNA can keep the formation of this Dox-inducible region, so making the entire cassette is essential for PKR, for the regulation of PKR activation. Yeah.
Okay, actually, I have a question. So you show that overexpression of circular RNA can repress PKR, right? But is there properties in terms of size or double-strand length, etc.? Or does any...
Yes, so basically double-strand RNA, if longer than 33 base pair, that can definitely stimulate PKR activity because they're going to provide enough room or space allow two PCAR binding at the same time to form the dimer. So if PCAR binds to snoRNA as a monomeric form, and they're going to keep it inactive. So the main point of this short snoRNA within the PCAR or within circRNA is to keep the PCAR as a monomeric as form.
Okay, thank you. Okay, another question. Knockdown of circRNAs affects proliferation and cell growth. I'm wondering if the mechanism is linked to microRNA release.
No, so for this circRNA does not associate with any microRNA.
Okay, and then is the expression of the oncogenic circRNAs, such as the circFAM 128, upregulated in cancer versus normal tissue? So I think Ram is asking about the expression of the circRNA upregulated. Is it upregulated in cancer versus normal tissue? Also, have you looked at pull-down mass spec to see what other proteins may interact with the circRNA and how they can influence tumorigenic phenotypes?
That's a great question. We didn't dive that far in terms of the physiological related to the tumorigenesis. So basically, we use this human cell line to address this question, largely focus on molecular mechanism. So at the moment, there are not enough data sets allow one to analyze the patient-derived circular RNAs. But for this particular one, we have found this high or increased expression in colorectal cancer, for example. So this is for the first part of question. For the second part of question, we didn't work on circular RNA pull down. And again, because it's very difficult to discriminate the circular RNA versus linear with that particular exon. So we're able to get, proteins interact with both circular RNA and, this exon region of the linear RNA. So we therefore decided to look at the gene-use eclipse data to look at, whether that cross back-splicing junction sites, what RNA binding protein associated with the circular RNA. So that dig out the adjective2. Yeah.
And then are M6A modifications on circRNAs important? Are important for PKR inhibition?
So this is basically two parts of the work. So in the first part, we are only talking about the unmodified circular RNAs, but probably because given the early study in Harvard's lab that and the Chen Ling lab basically. So basically, your M6 modification can change the structure of the RNA folding. So if that's the case, it's definitely possible mRNA circular RNA modification by M6A would be able to alter RNA forming capability, therefore affect the PCAR binding. But we didn't give a try modify circular RNA, then incubate with PCAR.
Okay. Do you think circRNA may repress PKR activation via producing peptides or other mechanisms besides structure-based regulation? Also, do you know any RBPs which help to maintain, alter the internal structure of circRNA in the circRNA PKR pathway?
Okay, so these are good questions. So for the first question, as I mentioned that, because in order to discriminate circular from linear RNA using shape map, we intentionally only worked on this short RNA, less than 500 nucleotides. So that means we can only do one set of divergent PCR to do that. So for that short circular RNA, that normally won't able to translate to protein because this iris sequence can at least more than even 300 nucleotides long. So that depends the possibility to make the small circle to be translated. So for that question is fine. So for the second question, what was that? PKR?
So do you know any RBPs which help to maintain, alter the internal structure of circRNAs in the circular?
That's a great question. That's a great question. That's something we're trying to dig out. We also wanted to learn the tertiary structure of circular RNA. So that's why we developed different methods to make the in vitro circles. Hopefully in the future, we're able to use other methodologies, including, or not excluding CRA-EMA EM to look at the circmRNP interactions. Yeah, but at the moment, I don't have the answer.
What is known about the immunogenicity of circular RNA and whether pseudouridine is able to reduce circular RNA immunogenicity like in mRNAs?
I think so. I think nobody gave a try, but it's definitely possible uridine lesion may dampen the RNA with immunogenicity. So generally speaking, so basically, I think the Grace Chen and the Harvard publication definitely can repeatable with the TD intron. We're able to see CCAR can induce eventual synthesis CCAR can induce immunogenicity. But I think it depends on your controls. So Anabana appears to have a lower level of immunogenicity. Also, now we're not going to see immunogenicity free. We see minimum immunogenicity. So it's very hard to define immunogenicity without using the animal model. This is on cellular level. Yeah.
Have you compared the RNA structure difference between circular RNA and its parental or linear RNA?
Yes, we did. The paper was published in 2009. We intentionally compared the sectional structure between the circular and the exactly circle-forming exons inside the cell. If I have some, let me show this. They're very different. For example, so this is a circle RNA, right? It can form something like this, but as the linear RNA, the same circle-forming region may pairing with distal region in the linear RNA. So think about the polymerase transcription. So as soon as the new RNA is made, the next RNA can form different folding. So you is definitely expert on this, but you can just with imagination, we're able to think about that. But once forming the circle, okay, it's become more rigid and unallow the pairing inside the circle. So making it very different from the linear RNA. Yep.
And do the circular RNA share some common sequence or prefer some sequence?
No.
I think the question is
no, because they came from exons. So that actually come from another similar question. Some people ask the circular conservation. I would answer the question like, based on what type of conservation you would define. If at a gene level, so one particular gene in mouse and human, they can both form the circular RNA, but that can form from exon 1 to 3. Circular RNA not came from exon 1, it came from after the first exon. So the circular RNA came from in mouse could be 2 to 3 or 2 to 4. However, in human, they can come from the same gene, but from the exons 5 to 7. So that kind of conservation really depends how you define them. So I would say the circular RNA, we define circular RNA conservation would came from exact same gene, the same exon.
And the last question, we have designed and validated antisense oligos that can either promote or inhibit circular RNA biogenesis. Do you think this will be a useful tool for circular RNA functional studies?
Of course. Yeah, it's exciting. Yeah, I would say, when I studied circular RNA, I had two low wave about this entire field. The first came from in 2015, before I published that 2016 Cell Reports paper. We studied how nascent circular RNA coupling with the protein transcription, and we found the circular RNA back-splicing efficiency is less than 1%. The most abundant circular RNA, less than 1% of the linear RNA splicing efficiency. I was very disappointed about that finding because, oh my God, we got something new and this such low levels. And later on, I found that have to be that in that way, because otherwise, if circular is efficiently made, the entire cell gonna have too many circular RNAs. So they have to be kept at a very low level. And then later on, we found it can be an excellent group of the circular RNA to modulate the immunogenicity, the homeostasis. Slowly produced and fastly degraded. So they had to be associated with the biogenesis. And secondly, came from how to study circular function. And we have no way to disrupt circular without affecting linear RNA until we use CRISPR-Cas9. So now I'm happy about this thing. I think also it provided another definitely useful and promising tool to study the circRNA, but how to be, be careful about the controls. You have to validate the linear RNA as well when you analyze the circRNA expression. Thank you.
Okay, thank you so much, Ling Ling, for the wonderful talk and taking all the time to answer all of the questions. So I know that we're running over time, so thank you for your time, and thanks everyone for joining the seminar. Thank you.
Thank you.
Bye.
Bye.